All right, so we've seen one way of doing a Hardy-Weinberg type problem, and that is, what do you do when you have genotype frequencies? We were able to do this one because we knew how many people in a population had each one of the three possible genotypes. There's another type of problem where you're not told what the genotype frequencies are. And problem one in this problem set is a great example. And it's data from a study of a Native American tribe in central Arizona called the Hopi. Now, this is going to revolve around Hardy-Weinberg, so think about this for a minute. How likely are the Hopi to be in Hardy-Weinberg equilibrium for any given gene? Well, first of all, this particular study looked at a population of about 6,000, so that population is pretty large. Okay, with me there so far. So on count one, it's pretty big. Migration in or out, the Hopi tend to keep to themselves. They have maintained their tribal culture pretty strongly. They're still, the Hopi language is still spoken. The old religion is still practiced and understood. They're not particularly hostile people. In fact, Hopi means the peaceful ones. But these days natives do tend to move off reservations for work and things like that. There are large native populations in major cities, for example, but not an awful lot of people marry in. They're not exactly hanging out signs saying, "Hey, come join us, be Hopi." That's not the way that works. So there might be some migration out, but there is little migration in. There was probably less in the 50s when this study was done. So it's large. They don't have much migration in or out. They're also a cultural isolate like the Dunkers. There is, let me see, mutation. Again, we can often leave that out because by itself, mutation usually does not push a population out of Hardy-Weinberg. That leaves natural selection and random mating. And it's possible that either one of these could play a role because this is a study of the incidence of albinism among the Hopi. Now think about this. The Hopi live in the desert of central Arizona. They are absolute wizards at dryland farming. They've got remarkable techniques for getting a harvest of corns, beans, and melons out of places where you wouldn't think you could grow a bleeping thing other than gravel. They're very, very good at what they do. But it does involve working out outdoors in the sun an awful lot because that's what you do if you're a farmer, right? You know why farmers are such great people? Because every farmer is outstanding in his field. Get it? Outstanding in his field, outstanding in his field. Okay. So what are some problems going to be if you are an albino? Yeah, if you spend too much time outstanding in your field, you're going to get severe sunburn and quite possibly skin cancer. Yes? Okay. So there could be natural selection working against albinism. If being an albino on Hopi land makes you less likely to live, or at least less likely to live long enough to have kids, then maybe selection would be making the allele frequency go down over time, right? Seems reasonable. Now, the Hopi not being stupid, I'm sure they've worked out that you can spend more time in the sun if you cover up with, loose-fitting clothing or something like that. So, people use culture all the time to circumvent what natural selection would otherwise do. So maybe that's happening. What that leaves is random mating. So how might that come into question? How might that be violated?
Maybe they avoid people that are albino, like refuse to mate.
Okay, yeah, there could be sexual selection against albinos if they're perceived as being unattractive or cursed or something like that. I don't know about the Hopi, but that's a problem in, there are parts of Africa where people born with albinism are likely to get killed. Yes.
It doesn't have to go that far. You just maybe they value work ethic and they see people with albinism and their ability to not be able to work as much as people that have the wild type.
Okay.
As not being attractive because they don't have the perceived work ethic.
Okay. It's possible that people who are albino among the Hopi, if they can't work outdoors as much, then maybe they're not valued as mates because they won't be able to support a family as well, or they might be perceived as not having a good work ethic or something like that. Possible. I hasten to mention that I don't know Hopi culture well enough to evaluate whether either one of those is true. But it is something we might have to consider at some point. I have actually heard it said that the opposite is true, that albinos are actually valued as mates because they are thought to have some kind of special power. Again, I hasten to mention that I should not be telling you things about the Hopi when I don't know anybody that's Hopi and I've never really asked. So I'm not trying to tell you what their culture is like because I don't directly know. I only had one chance once to drive across the Hopi reservation, and I was told not to do it because the tribal police hand out speeding tickets like you wouldn't believe for anybody that has out-of-state plates. It's worse than Damascus, Arkansas used to be. Or was the UCAPD bad about that? I mean, well, in that case, I know why. It's because like 20 years ago, I want to say it was like 2003 or something like that, we had this epidemic of students getting run over. And yeah, there were like 4 students got hit by cars in the space of a couple of months. I saw one of them happen. This guy's just walking across the road and this car just comes in and plows into him. I don't think it did any permanent damage, but it knocked him down and he just was barely able to get up and stagger over to the sidewalk. And that's about the time when they started putting in the speed bumps and the crossings, and I think they started cracking down on speeding on campus. But yeah, Damascus, Arkansas was so bad at handing out speeding tickets on Highway 65 that I think like the state Supreme Court basically had to tell them, hey, knock it off. I still go 5 miles below the speed limit whenever I'm going through Damascus. If you see a bright light on the road to Damascus, it's not God saying, why do you persecute me? It's the PD. Right. So yeah, I hesitate to conclude anything because I don't know Hopi culture very well. But if you are seriously studying Hopi genetics, those are things that you might need to find out about. So how do we work this problem? Here's the thing. Wolf and Ducapo found 26 albino individuals out of 6,000 Hopi. Okay, that's actually surprisingly high. And the reason why we can't crack this problem exactly the way we did with the Dunkers is albinism is controlled by a single gene with 2 alleles. Albinism is recessive, normal skin coloration is dominant. So why can't you calculate allele frequencies just from this? Like, you can tell who's albino, and the albinos are going to be homozygous recessive. Yes. Yes, you can't tell the difference between carriers and homozygous dominant people. You can't tell the difference between homozygous dominant and heterozygotes just by looking, right? They're both going to have darker skin, right? Yes? So right away, since we don't know all of the genotypes, we can't solve this problem the way we would a problem with the Dunkers, because you're not being given the genotypes. What we can do is assume that the population is in Hardy-Weinberg, and on that basis, we can try to estimate the frequencies of the genotypes. Now, our estimate is only going to be as good as our assumptions. If the population is not in Hardy-Weinberg equilibrium, then our estimates are going to be off. I've given you some reasons why the population might be in Hardy-Weinberg, but there are reasons to think that the population might not be in Hardy-Weinberg. There could be natural selection against albinos. There could be sexual selection either favoring or opposing albinos. We don't know, but if we momentarily suspend disbelief and assume we're in Hardy-Weinberg, we can calculate some estimates for the genotype frequencies. And then what we could do, if we wanted, and if the Hopi would let us, and I think they tend to be a little bit suspicious of, scientists sneaking around with big needles, and they're not necessarily wrong. But in theory, what we could do is then maybe do some genetic testing and try to work out what the real genotype frequencies are and compare them with our estimate of genotype frequencies. That would be easier to do now that we have DNA testing. It would not have been possible in the 50s when Wolf and Ducapo did their study. But let's take this as far as we can go. We'll calculate the expected allele and genotype frequencies if the population is in Hardy-Weinberg. So I'm going to hit the lights, and let's go to the, let's go to the whiteboard. Share whiteboard and clear all of this. So we know that we have 6,000 Hopi in the population that was studied by Wolf and Ducapo. 26 albino. And albinism is caused by a recessive allele. So the albinos have a genotype that I'll just write as albinom. Sorry, when the light is down, I can't see the keys very well. So there's 26 people with albinism. Genotype little a. If there's 6,000 Hopi and 26 albinos, there are 5,974 who are not albino. Genotypes homozygous dominant and heterozygous. Okay, right, you're with me there. Okay. If, that's a big if, for you. Draw some arrows to emphasize that big if. Okay. If we are in Hardy-Weinberg equilibrium conditions, then the frequency of the homozygous recessive genotype is equal to Q squared. So we're going to let P be frequency of the dominant allele. And we're going to let Q be the frequency of the recessive allele. Okay, with me there so far. If we are in Hardy-Weinberg, then the frequency of the homozygous recessive genotype, little a, is equal to Q squared. And it just so happens we know the frequency of the little a genotype. It happens to be 26 out of 6,000. So if we're in Hardy-Weinberg, then Q is equal to the square root of 26 divided by 6,000. There's the square root. There's the radical sign right there. Okay, that's kind of a mess, but you can tell what that is. Okay, you see what I'm doing there? Yes? Да? Все хорошо? Отлично. So Q, therefore, is going to be equal to what? What's the square root of 26 divided by 6,000? 0.066. Hang on. 0.066. Okay, you with me there? What's the frequency? What's P? What's the frequency of big A? You could do it the agonizingly painful way, and you could say that the frequency of homozygous dominant and heterozygotes is 5,974 divided by 6,000. And that's also equal to P squared plus 2PQ. So you could say P squared plus 2PQ equals 5,974 divided by 6,000. And you solve that by substituting 1 minus P for Q. So you could say P squared plus 2P minus P squared plus, okay, you substitute and you solve it as a quadratic, but we don't need to do that. You don't need to go the long way around. And because we know that P must equal 1 minus Q, and 1 minus 0.066 is 0.934. All right, am I making sense there? Okay, you could go around and do it the long way, but why? Because we know that if there's only 2 alleles, their frequencies have to add up to 1. Yes? Okay, great. So now what do we do? Well, if we are in Hardy-Weinberg, then the frequency of homozygotes is p squared, dang it, equal sign p squared. And that's equal to 0.934 squared. So 0.934 squared is going to be like 0.82 or something. What do we got? 0.934 squared. 0.872. 872. All right. That up a little bit. And the frequency of the heterozygote is going to be equal to 2pq. And what's 2pq? Well, it's going to be 2 sorry, I've got to move that out of the way. Frequency of heterozygotes is 2pq. That's 2 times 0.934 times 0.066. Sorry, which is 0.123. One, two, three. Again, did I mention that I cannot really see the keys very well unless I stare at them? Okay, so we basically worked the problem. If the population is in Hardy-Weinberg, then the frequency of homozygous dominance would be 0.872, or 87.2% of the Hopi would not be carriers of albinism. And then 0.123, 12% of the Hopi, that's roughly what, 1 in 6, would be carriers of the albinism allele. That could be interesting to know that we have a surprisingly high frequency of carriers. It's an order of magnitude less than this in most human populations. So that's our problem. That's the solution. Again, our solution's only as good as the assumptions that went into it. And for some reasons we've already gone into, the Hopi might not be in Hardy-Weinberg equilibrium. It is also possible that 2 of the Hardy-Weinberg conditions could be violated, but the violations are canceling each other out. Maybe albinos have greater chance of dying, but that's made up for by the possibility that the albinos are seen as powerful and people want to mate with them. So it's possible we could have two violations, but they're working in opposite directions. Again, and I think I'm saying this repeatedly because even though nobody outside this class is likely to see it, I am going to post these on YouTube. And you know, this is public. I do not claim to speak for the Hopi. I don't know their culture well enough to know which is true, to know what's going on, but this is as far as we can get with the data that we actually have. And it's the kind of thing that you might do sometime if you're investigating a population that's got an unusual allele frequency, possibly of something that causes a genetic disease. It wouldn't surprise me if the geneticists who were studying Tay-Sachs syndrome among the French-speaking population of Eunice, Louisiana, might have done something similar to try to give a rough estimate for how many people might be carriers. That's the kind of thing that sometimes you might need to know. Okay, clear? Everybody super happy here? Okay, let me talk for just a little bit. Instead of working a problem, let me talk for a second about why Hardy-Weinberg works. Now, you can demonstrate this for any population you like. It doesn't specifically have to be quite the way I'm going to do it, but I'm going to do it this way. There we have the ocean floor. All right, I will demonstrate that it is the ocean floor because way up above, of course, we have there's ocean waves. There we are. There's some ocean waves, and we can do oh, yellow's a nice color. Now let's do green. Here we have a nice little sailboat. And there we've got some people who are waving because they're friendly and they own a boat. And they want us to know that they own a boat. Okay, what the heck? We'll put some color here. We'll put some yellow and red stripes on the sail. Come on, Wagner, get to the point. There's a nice little sailboat up there. On the bottom of the ocean, let us say, we have oysters. All right, that's my attempt at drawing some oyster shells. And here's why I picked oysters. When the moon is in the right phase and the tides are high, in summer, oysters reproduce basically by firing off great big clouds of either eggs or sperm into the water. This is one of the reasons why you don't want to eat oysters gathered in summer. It's because they've spent so much metabolic energy on producing lots of eggs and sperm that they don't have very good flavor and they're kind of watery. The other reason is that's when they're most prone to be carrying things like paralytic shellfish poisoning toxin, things like that. You guys know that rule? You only eat oysters in months with the letter R. So you don't eat them in May, June, July, or August. But in September, October, right, January, March, April, you can eat oysters. Okay, the summertime is when they spawn and they don't taste very good because they put so much energy into releasing ridiculous numbers of eggs and sperm. And I'm drawing oyster eggs and oyster sperm here. Not to scale, they're not really that big. Oyster eggs are not really the size of beach balls.
Or boats.
Or boats. Yeah, oyster eggs are pretty much microscopic. But yeah, that's pretty much what the sea would look like. It's freaking oyster gamete soup when the oysters are, hot to try. When you're a clam and you have very little brain and no head and you're permanently glued to the ocean floor, this is the way you do it. And imagine that we've got some gene that oysters have. It doesn't really matter what that gene is. It's something or other. Every oyster's got 2 copies of it. So I'm just going to call it gene A, and it's got 2 alleles that we can write as big A and little a. So some of these oysters, like this one, are going to be homozygous dominant. Maybe this one is homozygous dominant. Maybe that one is. And some of these oysters are going to be heterozygotes. Maybe that one is, and that one is, and that one is, why not? And some of these oysters will be homozygous recessive. So that one can be, and that one can be, and that one can be, and that one can be, whatever. All right, so our oysters all have genotypes. And our oysters all fire off massive clouds of eggs and sperm into the water. Since the sperm and eggs are, of course, haploid, some of the eggs are going to be carrying a big A allele, and some of those eggs will carry a little a allele, and some of our sperm will be carrying a big A allele, and some of our sperm will be carrying a little a allele. Okay, you with me there? All right, and sperm and egg are going to meet pretty much at random, right? The gametes are released by the oysters. They're going to get churned up by the currents a bit. So it is, as for all intents and purposes, it's random whether an egg is fertilized by a big A sperm or a little a sperm, right? Yes? Okay, so gametes are pairing up completely at random. Okay, think about this. Assume, to make things simple, that the allele frequency isn't different between eggs and sperm. There are cases where it could be. If we had a sex-linked gene, let's say, then there might be an allele that is not carried, that is carried in potentially all of the eggs, but only half the sperm or something like that. That gets a little more complicated. But assume right now there's no difference in allele frequency in eggs and sperm. Now we know, and half a tick, I think I'm going to shrink the font size just a little. So we have the frequency of big A is some number between 0 and 1, and we're going to call that P, right? And the frequency of little a is some number, and we're going to call it Q. And the sum of those two is equal to 1. That should be a plus sign. Okay, yes? All right. The frequencies are also probabilities, right? The frequency of big A alleles is also the probability that if you pick a gamete out of the soup at random, it's going to be carrying the big A allele, right? And the frequency, the Q, is also the probability that if you pick a gamete at random out of the soup, it's going to be packing the little a allele, right? Yes? Okay, so that sets up a probability equation. The probability of two things happening together is the product of the separate probabilities, right? Right, yes? The product of, okay, the probability of getting 2 heads when you flip a coin is the probability of getting 1 head multiplied by the probability of getting 1 head, right? And if it's a fair coin, those are both 50-50. So the probability of getting 2 heads is 0.5 times 0.5, which is 0.25, 1/4, right? So let's say that I reach in and I pull out an egg and a sperm. What is the probability that both of them are going to be carrying the big A allele? Well, the probability that we're going to get that the egg and the sperm are both going to be carrying the big A allele, and they'll give rise to a homozygous dominant big A zygote, is simply equal to P times P, which is P squared, right? And the probability that if we pull 2 gametes at random, and they're both going to be carrying the little a allele, is simply equal to the probability that the first gamete is packing a little a times the probability that the second gamete is packing little a. So that's just Q times Q. You with me there? All right. What happens if we have to calculate the frequency of heterozygotes? Well, we actually have to do that in more than one way. Dang it. Backspace, close parentheses, there. Close parentheses. Put the equals key right next to backspace. All right, there's two ways that could happen. One way is we could reach into our gamete soup and pull out an egg, and the probability that the egg has big A is P, right? And then we reach in and we pull out a sperm, and the probability that the sperm has little a is Q, right? That's the probability of reaching in our soup and pulling out a big A egg and a little a sperm, right? That probability is PQ. And that, if we fuse that egg and that sperm, we get a heterozygote, right? Yes? But is that the only way we can get one? What's the other way? Yeah, we could also, the other possibility is we could reach into our gamete soup and pull out an egg that's got little a, and the probability of that happening is Q. And then we could reach in and pull out a sperm with big A, and the probability of that happening is P. So the probability that two randomly chosen gametes, one will have big A and the other will have little a, is the sum of the probability of the two separate ways that could happen, right? Either the egg is big A and the sperm is little a, or the sperm is big A and the egg is little a. And in this system, we can't tell which is which. So the probability that any pair of gametes is going to give rise to a heterozygote is equal to P times Q plus Q times P, or in other words, 2P squared. Right? Did everybody see how I did that? Yeah, 2 times P, yeah. Well, it was PQ plus PQ. So that's 2 times P times Q, 2P squared. 2PQ, not 2P squared. Oh, good grief. I definitely picked the wrong week to quit crystal meth. Yeah, 2PQ plus PQ is 2P squared. I might have to delete that bit from the recorded version. People are going to think I'm an idiot. Thank you for setting me straight. Okay, that's why the Hardy-Weinberg equilibrium works. Okay, that's why we get those terms. You can derive it from species that don't mate in this way. It takes kind of a while, and I'm not going to attempt it, but we could do a similar derivation if we had, a population that instead of firing gametes off at random, courted and mated in a much more discrete fashion that you're likely to be familiar with. We could derive this as well. It doesn't really matter by itself what the mating system is for the species that we're wanting to work on. Okay, you with me there? All right. Hey, let's see how this would work. Oh, we have a on the chat. Why do we not multiply the population by 2? Oh, like we did on the other, because in this case, we're not calculating allele frequencies among the oysters down here on the bottom. We're calculating allele frequencies among the gametes, each one of which only has one copy of each allele. So we're calculating the allele frequencies among the gametes. And if we start selecting gametes randomly, then when the gametes pair up and produce the next generation of oysters, we're going to get Dang it, that's not what I wanted. We're going to get p squared, 2pq, and q squared as our genotype frequencies in the next generation, regardless of what big A and little a, regardless of what p and q actually are. Okay, that makes sense. Thank you. I try to make sense. Sometimes I does it, and sometimes I doesn't. Okay. In the time we've got left, let me show you how we can apply this to cases where we've got more than 2 alleles. The practice problems for this are actually at the end of the set, and we will work one. But let me go over a couple of things first. To do this, I have to erase all these letters. Let's say that our oyster population here seem to have a seem to have a headless sperm right here. Let us say that our population of oysters has some gene, but instead of 2 alleles, it's got 3. And it doesn't matter for our purposes which one is dominant and which is recessive or what the situation is. But let's say we have some oysters that are homozygous for allele 1, and I'm just going to write this with little subscripts, and some that are homozygous for allele 2, which we'll just write A2, and some might be homozygotes for allele 3, and some might be homozygotes there A1, A2, and this one might be a heterozygote for A2, A3, and this one might be a heterozygote for A1, A3. We've got 6 possible genotypes, right? Yes? You with me there? Okay, 6 possible genotypes. And they fire off their massive clouds of eggs and sperm into the water, and the same thing happens. Some of the eggs are carrying A1, some of the eggs are carrying A2, some of the eggs are carrying A3. And some of the sperm are carrying A1, and some of the sperm are carrying A2, and some of the sperm are carrying A3. And I apologize again for my rotten penmanship, but here we've got a situation where we've got 3 alleles. You with me there? Okay. We can work out how to solve 3 allele problems exactly the way that we did it with the 2 allele problem, right? So we can assign frequencies to each one of these alleles in the pool of eggs and sperm that's floating around. So we could say that we could let P be the frequency of the A1 allele, whatever that is. And we can let Q be the frequency of the A2 allele. And we could pick another letter, completely at random. Let's just say that R is the frequency of the 3rd allele, the system, which we'll call A3. The pros would probably just use P sub 1, P sub 2, P sub 3, P sub 4 for however many alleles they were working with. In humans, there's at least one gene that I think has 26 alleles. So if you ever were so unlucky as to have to work on that one, you just use subscripts to keep them apart. But we can go ahead and use P, Q, and R. Okay. Now the probability that we reach in and get a gamete with A1, and we reach in and we get another gamete with A1, is equal to P squared. Yes, right? Same reasoning that we've used earlier. There, finally got it to look right. Yes? The probability of getting an A1 homozygote is the probability that a randomly selected egg is going to have the A1 allele and a randomly selected sperm will have the A1 allele. All right, and by the same reasoning, the probability that a random pair of gametes will both be packing the A2 allele is equal to the product of the probabilities of any gamete having the A2 allele. Yes, Q squared. And the probability that any two randomly selected gametes will both be packing A3 is going to be equal to R squared. Right? Yes? Okay. What that means is that the frequency of homozygotes in the population is going to be the sum of the squares of all of the allele frequencies. Right? Okay, the frequency of homozygotes in a population, all homozygotes, is equal to the sum of the squares of all the allele frequencies. So if we have 3 alleles, then the frequency of all homozygotes is going to be P squared plus Q squared plus R squared. If we had 4 alleles, the homozygotes in Hardy-Weinberg would be P squared plus Q squared plus R squared plus S squared. If we had 5, it's P squared, Q squared, R squared plus S squared plus T squared, as far as we want to take that. Okay, with me there. All right. And then, of course, the probability of the heterozygotes. Say the probability of the A1, A2 heterozygote is going to be the probability that a random egg will have A1 and a random sperm will have A2. That's P times Q. Plus the probability that a random egg will have A2 and a random sperm will have A1. And that's P times Q. Q times P, but multiplication is commutative. And that's going to be 2PQ. And we can use the same reasoning to show the probability of A1, A3 heterozygotes is going to be 2PR. And the probability of an A2, A3 heterozygote is going to be 2QR. Okay, did everybody see what I did there? Okay, just remembering this, remembering how you derive this means that you can easily expand Hardy-Weinberg problems to cases where you've got more than 2 alleles. And we very often do have more than 2 alleles. And to show you how this plays out, let's go look at a practice problem. We'll switch over to practice. And you go down to the end of it. Do this one. Okay, let's first do 12 and then do 11. Oh yeah, this is one of Jim Valentine's old papers. One of the authors of this study was one of my professors in grad school back in the day. So 1973, Francisco Ayala sampled a population of deep sea starfish. It's not easy to go down 1,244 meters to observe these starfish in their natural habitat. So I assume they dredged them off the sea bottom. A dredge is kind of like a big net scoop that just scrapes things off the bottom. And when, in our previous problems, those come from the 50s. By the 1970s, a chap at Harvard by the name of Richard Lewontin has invented gel electrophoresis. You guys all got to do gel electrophoresis at some point, yes? Right, that's where you put samples of, it could be DNA or protein, depending, into a little well on an agarose gel, and then you put the gel in a rig that applies electric current to it, right? And the molecules move through the gel. Okay, well, they did this with tissue fluid from these starfish, and they looked for one gene in particular, an enzyme, it doesn't matter what it is, it's called tetrazolium oxidase. And it turns out that that one gene has 4 alleles, which you can abbreviate TO 100, 107, 114, and 120. Those numbers, I think, just refer to how far the protein moves in the electrophoresis chamber. Okay, so if a starfish is homozygous for this first allele, TO 100, when you electrophorese it, it will have one band, let's say it's 100 millimeters away from the start. One that's heterozygous for 100 and 107 will have 2 bands at 100 and 107 millimeters from the start, and so on. So this actually allows you to detect genotypes directly instead of having to guess at them. And the data that they published is this set of allele frequencies. The frequency of TO 100 is 0.206, TO 107 is 0.147, 0.353, and 0.294. They observed that in this population, 70.6% of the population was heterozygous. Does that mean that we're in Hardy-Weinberg equilibrium? And your brain may be seizing up right now, but this is actually quite easy. This is an easy problem. Let me go back to the whiteboard and maybe not do this one at full screen size so I can see what I'm writing. I'll shrink you. Okay, shrink that a little smaller and erase everything on it. Oh, clear all drawings. Okay. So here we're told that the frequency of the first allele, which is the frequency of TO-100 equals 0.206. We'll let Q be the frequency of TO 107. close parenthesis. Backspace, close parenthesis equals 0.206. Okay. Sorry, again, I can't see the keyboard clearly. And that's equal to 0.147 height. And we can call R is the frequency of the TO114 allele, which is 0.353. And S is the frequency of the TO120 allele, and that's 0.294. Okay, great. What we want to know is whether the population's in Hardy-Weinberg equilibrium or not. Oh, we also know that the frequency of heterozygotes is equal to told us what it was. I'm trying to move it out of the way so I can see this. Okay, 70.6. So the frequency of the heterozygotes is 0.706 or 70.6%. Okay, with me there so far? The easiest way for me that you could do this is to remember that since every starfish is either heterozygous or homozygous, the frequencies of both have to add up to 1. Hi, Carl. So the frequencies of both must add up to 1. And so the frequency of the homozygotes is equal to 1 minus the frequency of the heterozygotes. So that's going to be equal to 0.294, right? Yes? Now the frequency of the homozygotes is easy to calculate because the frequency of the homozygotes is equal to the sum, sorry, there's my attempt at drawing a sigma. There we go, there's a little sigma of the allele frequencies squared. Actually, there needs to be an indexing right there. Get rid of that. We'll let every allele frequency be P1, P2, P3, so far. So allele frequencies we can just index as Pi. And so the frequency of homozygotes is just going to be the sum of the squares of all of the allele frequencies. So you just have to calculate 0.206 squared plus 0.147 squared plus 0.353 squared plus 0.294 squared. And presumably somebody's got a calculator and can plug and shove that really quickly. You have these 4 numbers, just square each one and add the squares. .275. Anybody else doing that? Did anybody else get that?
Sure.
I'm not saying you're wrong, but you know, it's always good to check these things. Okay, you got 0.275. Anybody else got that? You got it too? Is 0.275. Okay, that's the actual frequency. Or wait, that's the expected frequency if we're in Hardy-Weinberg. And the frequency of the heterozygotes is equal to 1 minus that, which is going to be 0.725, right? Yes? Okay, so draw. Okay, so that right there is our expected if the population's in Hardy-Weinberg. If it is, we should have 0.725 heterozygotes. Right over here, that's our observed. That's the actual number of heterozygotes that there are in the population. And you can see they're close. They're not identical. And it's possible that this population is not quite in Hardy-Weinberg equilibrium. We could do further testing to try to see if this is true for all alleles or just this one. But yeah, we've solved the problem. So you can expand this Hardy-Weinberg relation to any number of alleles that you want to. If you just remember that the frequency of homozygotes of all kinds is just going to be the sums of the squares of all allele frequencies if your population is in Hardy-Weinberg. And if it's not, you know something interesting is going on. We can't directly study all those starfish, or we could, but it's kind of hard. But we can at least say that if the population is not in Hardy-Weinberg equilibrium, then the population might be small, or there's migration, or there's non-random mating, or there's natural selection, or there's mutation. We know that something interesting is going on in our starfish, even if we don't directly observe the starfish in their natural habitat. Okay, everybody happy with that so far? You feel like you've got so much knowledge that it's practically dripping out of your ears. Okay, good on you. Go ahead and take another break. We'll reconvene at 11:40, and I'll try to finish off with something short. But for now, I will